By now, dormant viruses have almost certainly reactivated inside their bodies. Their bones have been dissolving for 10 days. Their hearts have begun to reshape. The 4 crew members who splashed down in the Pacific 4 days ago walked off the recovery ship, smiled for the cameras, and looked completely fine. None of what is happening inside them is visible from the outside. This is not the story about the heat shield or the 6 minutes of radio blackout. This is the one nobody is reporting yet. What 10 days in deep space at radiation doses 18% higher than anyone had planned, with particles that no wall can stop did to the bodies of Reed Wiseman, Victor Glover, Christina Koch, and Jeremy Hansen. Inside a Harvard laboratory right now, tissue grown from the actual blood cells of these 4 astronauts is being analyzed gene by gene. Some of what it reveals may never reverse. The damage started before they cleared the Van Allen belts. To understand why 10 days matters, you need to understand where those 10 days were spent. The International Space Station orbits at 400 kilometers above the surface. That number sounds enormous, until you consider that Earth's magnetosphere, the invisible magnetic bubble generated by the planet's molten iron core, extends roughly 65,000 kilometers in the direction facing the Sun. The station orbits nearly 200 times below the bottom edge of that protection. Every astronaut who has lived aboard it for the past quarter century has been shielded, imperfectly but meaningfully, from the worst of what open space can deliver. Artemis II left that protection behind within hours of launch. The spacecraft crossed both Van Allen radiation belts on the way out and crossed them again on the way back. The inner belt, a concentrated band of high-energy protons trapped in Earth's magnetic field, beginning at roughly 1,000 kilometers altitude, bombards unshielded tissue at rates that would be lethal over hours of direct exposure. Orion's walls bring the acute dose during the crossing down to a manageable level, and the transit through the most intense regions lasts approximately 30 minutes each way. But the crossing happens twice, and the accumulated dose during those transits is part of the biological accounting now underway in Houston. Between the 2 crossings, the crew spent 8 continuous days in deep space outside the magnetosphere, outside any meaningful protection from the 2 categories of radiation that define the biological threat in open space. The first category is solar particle events. Bursts of high-energy protons released by solar flares and coronal mass ejections. They can arrive with almost no warning, deliver dangerous doses over hours or days, and are at least partially manageable. The walls of Orion provide 7 to 10 grams per square centimeter of aluminum, shielding enough to attenuate lower-energy particles from a typical solar storm. Mission Control monitored the Sun around the clock throughout the mission, ready to direct the crew to shelter near the stowage bags and water supplies, where the material density is highest. The second category is what kept radiation physicists awake throughout the planning of this mission. Galactic cosmic rays (GCR) do not come from the Sun. They originate outside the solar system entirely, in the remnants of supernovae and the magnetic fields of neutron stars scattered across billions of light-years. They arrive from every direction simultaneously, at velocities approaching the speed of light, and they pass through the walls of the spacecraft, through the bodies of the crew, and out the other side without decelerating appreciably. They leave damage along the way, shattered DNA strands, ionized atoms, broken molecular chains, and then they continue. There is no shelter from galactic cosmic rays. Not in Orion. Not in any vehicle that can actually reach deep space. What makes GCR uniquely dangerous is their composition. Roughly 87% are protons, 12% alpha particles. The remaining 1% are heavy ions, carbon, oxygen, silicon, iron nuclei, accelerated to near light speed by stellar explosions. These are called HZE particles, high-charge, high-energy nuclei. When a medical X-ray ionizes tissue, the damage is scattered and modest, and the cell's repair mechanisms handle it routinely. When an iron nucleus moving at 96% the speed of light passes through a cell, it deposits a dense, continuous track of ionization, a column of molecular destruction, so narrow and so concentrated that the cell's repair machinery often cannot reconstruct what was broken. The track is not scattered. It is a wound. The Artemis II crew spent 10 days moving through that environment, every hour, without interruption. And preliminary data from the HERA radiation sensors now being analyzed at Johnson Space Center suggests the crew received approximately 18% more radiation than the pre-flight models had predicted. Carrie Lee, Orion's radiation health scientist at JSC, attributed the variance to the unpredictable nature of the current solar maximum. The numbers remain below acute danger thresholds, but the models were wrong, 18% more than planned, on the very mission that two scientists had publicly warned was launching at the wrong moment in the solar cycle. That gap between what the models said and what the sensors measured is the first thing the data from this mission will need to explain. The engineers who built Orion understood the threat. The radiation protection strategy was layered. Passive shielding in the capsule walls, 6 active sensor units called hybrid electronic radiation assessors throughout the cabin, personal dosimeters worn by each crew member, and a dedicated solar weather team at Johnson Space Center monitoring every fluctuation in the sun's output. The HERA units, built around silicon chips, originally developed at CERN, measure dose in real time, identify particle types, and can trigger audible alarms if levels spike above safe thresholds. The German Aerospace Center contributed an upgraded heavy ion detector, the M42 EXT. With 6 times the energy resolution of the instrument that flew on Artemis, the specifically engineered to capture the most biologically damaging particles, this instrumentation represents decades of refinement and constitutes the most precise measurement of the deep space radiation environment ever conducted with a human crew aboard. But instrumentation only measures the threat. It does not stop it. The most important radiation protection experiment aboard Artemis II was not a sensor. It was 4 devices the size of a USB drive, each containing living human tissue, loaded into a triangular container and stowed just before launch. The experiment is called AVATAR, a virtual astronaut tissue analog response developed at Harvard University's Wyss Institute for Biologically Inspired Engineering by Donald Ingber, the institute's founding director. Each chip contained microfluidic channels lined with bone marrow cells derived from the blood of one specific astronaut, not generic cells, not donor tissue. The actual cells of Wiseman, Glover, Koch, and Hansen, extracted months before the mission, converted through a multi-step laboratory process into induced pluripotent stem cells, then differentiated into bone marrow progenitor cells. The chips maintained those cells alive at body temperature for the full 10 days, circulating nutrients through channels narrow enough to replicate capillary flow, removing waste products, and replicating to the extent current technology allows the internal environment of a living human body's most radiosensitive tissue. Those chips flew the same trajectory as the same astronauts. They experienced the same radiation, the same microgravity, the same duration. An identical set remained on Earth as a control, maintained under identical conditions except for the one variable that cannot be replicated in any laboratory: the radiation environment of deep space. The bone marrow was not chosen at random. It is among the most radiosensitive tissues in the human body, the factory that produces every red blood cell, every platelet, every cell of the immune system. Lisa Carnell, director of NASA's Biological and Physical Sciences Division. Someone may be radio-resistant, and we will learn something new. Or maybe someone is extremely susceptible to radiation. Now we can design medical kits that we can personalize for their journey. We can send the right therapeutics with them to make sure they stay healthy. Donald Ingber, the experiment's principal investigator. This has never been done before, comparing the cells of an astronaut who stayed on Earth with the same astronaut cells exposed to deep space. If it works, We could study hundreds of people. We could separate what radiation does from what microgravity does. The chips will be analyzed using single-cell RNA sequencing, a technique that reads the expression state of thousands of Rydau genes inside individual cells simultaneously, producing a molecular portrait of how radiation altered what each cell was doing. The analysis takes weeks to months. The data are being processed now. What those chips will show is unknown. The point is that for the first time in human history, there will be an answer, not a model, not an extrapolation from mouse tissue irradiated in a ground-based accelerator, but an answer from the actual cells of actual human beings who went to deep space and came back. Those cells were changing from the moment the capsule left the magnetosphere. The crew's bodies were registering the same process through physiological changes that begin faster than almost anyone expects. Within seconds of reaching microgravity, approximately 2 liters of fluid blood and interstitial fluid that gravity normally keeps pooled in the lower body shifted upward toward the head and chest. Jeremy Hansen described it live on April 5th during a session with Canadian school children. "I'm sure my face is a lot puffier now because there's a lot more blood in the upper regions of my body. I can feel it in my head. It's not painful, but it's slightly different." What Hansen was describing was his cardiovascular system responding to the removal of the gravitational gradient it had calibrated against his entire life. The heart, suddenly receiving more venous blood than it was designed to handle from below, began adjusting not just functionally, but structurally. Research published in the American Journal of Physiology documented what that structural adjustment looks like. The heart becomes approximately 9.4% more spherical in microgravity, beginning within 2 weeks. The Artemis II crew spent 10 days there. The remodeling almost certainly began. After 6 months in orbit, the left ventricle can lose 8 to 12% of its muscle mass through disuse atrophy. The heart, like every other muscle in the body, shrinks when it is not challenged. The same fluid shift that is rounding the heart is also pressing against the back of the eye. That is not a coincidence. It is the same 2 liters of displaced fluid pushing upward against everything in its path. The extra pressure pools behind the optic disc, which swells. The globe flattens from behind. The lens shifts position. The syndrome has a name: Spaceflight-Associated Neuro-Ocular Syndrome, or SANS. It affects approximately 70% of astronauts on long ISS missions. Some return needing reading glasses for the first time. Some structural changes persist for years after landing. Whether 10 days in the deep space radiation environment at higher doses than any ISS mission is enough to initiate the early cellular signs is one of the clinical questions the post-flight evaluations now underway in Houston are designed to answer, and below the level of any eyeglass prescription. Bone. Biochemical markers of bone resorption, the process by which the body breaks down skeletal tissue, rise as early as day 8 of spaceflight. In the weight-bearing bones of the skeleton lumbar vertebrae, proximal femur, hip, the loss rate reaches 1 to 1.5% per month in microgravity. On Earth, a healthy adult loses roughly 1% of bone density per decade. In space, the rate is between 120 and 180 times faster. For the Artemis II crew, 10 days means the biochemical machinery of bone loss was fully activated, even if the structural change was not yet measurable on a standard scan. Simultaneously, in the same bodies, muscle fibers that exist specifically to maintain an upright human being against gravity received no meaningful mechanical signal. The soleus, the gastrocnemius, the intrinsic muscles of the lower back, all of them diminishing, quietly, for 10 days, while the crew photographed the far side of the moon. The flywheel device aboard Orion, 30 pounds, 400 pounds of maximum resistance loading, provided 30 minutes of exercise per day and slowed the atrophy. It could not prevent it. Studies from shuttle missions of comparable length documented losses of 6 to 10% in specific anti-gravity muscle groups even with daily exercise. 10 days is not 6 months. But the process starts immediately. And now, in a Houston laboratory, blood and urine samples from 4 astronauts are revealing exactly how far along that process had gotten when they hit the Pacific. The first biological snapshot of a human body at the moment of return from deep space in more than 50 years. Here is the number that has defined the fundamental argument over deep space human exploration since Orion was still on the drawing board. A complete Mars mission accumulates approximately 1 sievert of effective radiation dose. NASA's career exposure limit for astronauts is 600 millisieverts. 1 sievert is that limit, plus another 400 millisieverts on top. ESA, Roscosmos, and JAXA each set their own career limits, ranging from 500 to 1,000 millisieverts. Every one of those limits is exceeded by a Mars mission under current shielding technology. To send a crew to Mars under the rules that govern every other human spaceflight, NASA would need to grant a formal exemption from its own safety standards. Dr. Francis Cucinato holds more than 350 peer-reviewed publications on space radiation biology. He served as chief radiation scientist for NASA's Johnson Space Center for over a decade before joining the University of Nevada, Las Vegas. In 2021, he published a direct challenge to the agency's updated radiation limits in the journal Life Sciences in Space Research. His argument was not primarily technical, it was ethical. A 5-year-old female astronaut sent to Mars under the new framework could have a greater than 10% risk of dying within 15 to 20 years from radiation-induced cancer. He stated his conclusion plainly. "This is like playing Russian roulette with the crew," NASA pushed back. The agency argued that improved cancer risk modeling justifies the updated limits and that informed consent from the crew addresses the ethical dimension. Notice what that response did not say. It did not say Cucinato's risk projections were wrong. It did not say the career limits would not be exceeded on a Mars mission. It said the limits were updated and the crew would be informed. Those are not the same sentence. Cucinato was not alone. In February 2026, weeks before Artemis II launched, Dr. Victor Manuel Velasco Herrera of the Universidad Nacional Autónoma de Mexico published a predictive model of solar superflares in the Journal of Geophysical Research, Space Physics, identifying the mid-2025 to mid-2026 window as a period of elevated solar risk, and recommending explicitly that the mission be delayed to late 2026. "The exploration of the Moon," he wrote, "is not a race. The mission launched on April 1, 2026. His paper was not referenced in any of NASA's pre-launch press conferences. The preliminary dosimetry data now being processed in Houston will be the first objective test of whether his warning had merit. The 18% excess above predictions is not proof, but it is not nothing either. There is also a physics problem embedded here that almost no coverage has addressed. When a galactic cosmic ray heavy ion strikes the aluminum wall of Orion, it does not simply stop, it undergoes nuclear spallation, fragmenting into a shower of secondary particles, neutrons, protons, lighter ion fragments, gamma rays. Those secondaries can be more numerous than the original particle, and in terms of biological damage, more dangerous. Shielding strategies are largely ineffective for reducing galactic cosmic ray exposure, and excessive or poorly chosen shielding materials can actually increase effective dose rather than reduce it. Not inadequately shielded, cannot be shielded. With any vehicle that can actually leave Earth. 8 days, no shelter, no way out. You cannot see the radiation, you cannot hear it. You cannot feel the particles passing through the aluminum wall and through your chest and out the other side. The dosimeter in your pocket registers silently. The environment looks exactly like open space always looks, black, absolute, and indifferent. Nothing indicates that anything is happening. Then, in the darkness, with your eyes closed, you see a flash, not light. There is no light. It is an iron nucleus moving at 96% the speed of light, passing through the wall of the spacecraft, entering the back of your eye, ionizing the photoreceptor cells directly, triggering them the same way a photon would, through brute physical force rather than optical absorption. Your retina fires. A flash appears from nothing. Neil Armstrong saw it during Apollo 11. Buzz Aldrin saw it. Every crew that went to the moon reported it. Initially, NASA did not know what it was. Then it happened again on Apollo 12, then 13, then 14. By Apollo 15, James Irwin described one as a bright streak across the retina. David Scott reported one as blue with a white tone. Like a blue diamond, the only non-white flash ever documented on a lunar mission. By Apollo 17, Harrison Schmitt reported them as practically continuous during darkness adaptation. One flash every 2.9 minutes in deep space, compared to one every 6.8 minutes in low Earth orbit. A specialized instrument flown on Apollo 16 and 17, the Apollo Light Flash Moving Emulsion Detector eventually confirmed two direct correlations between high-charge cosmic nuclei traversing a crew member's eye and a reported visual sensation. The particles were visible, not as light, but as physics arriving inside the retina of a living human being. The Artemis II crew experienced the same phenomenon. Think about what that means for a moment. You are floating 252,760 miles from the nearest hospital, seeing light that does not exist because subatomic particles from a stellar explosion that occurred billions of years ago are passing through your skull and firing your visual cortex on the way through. There is no way to close your eyes against it. It is arriving from every direction simultaneously, every 2.9 minutes, continuously, for 8 days. Simultaneously, at the cellular level, something subtler was underway. Roughly 53% of all astronauts who fly missions of 10 to 16 days show reactivation of latent herpes viruses, Epstein-Barr, varicella zoster, cytomegalovirus viruses. The immune system keeps suppressed through constant T-cell surveillance. In the physiological stress environment of spaceflight, elevated cortisol suppresses that surveillance. The viruses sense the reduction in pressure and begin replicating. In studies by Dr. Satish Mehta and Dr. Brian Crucian at Johnson Space Center, viral shedding in astronaut saliva during short missions reached 53% prevalence compared to 2 to 5% in matched controls on Earth. The shedding is usually clinically silent, but the immune system is being outpaced. Saliva samples collected aboard Orion on special paper cards, standard refrigeration was not available in the capsule, are now being analyzed. The results will show whether the Artemis II crew was among that 53%, and if they were, those viruses were replicating inside them while they watched flashes of impossible light appear in the darkness behind their eyelids. Over the far side of the moon, Victor Glover is the first black astronaut in history to travel beyond low Earth orbit. He is a father. When the crew watched Earth slide in front of the sun from behind the moon, a solar eclipse no human had ever witnessed from that vantage point, he called it one of the greatest gifts of the mission. He was floating 252,760 miles from home, seeing something extraordinary, while cosmic radiation passed through his body at doses 18% above what anyone had planned for, and the cells in his avatar chip recorded every moment of it. That experience has been reported. What has not is what a laboratory in Houston is now reading in those cells. Each avatar chip contained the actual living cells of one specific astronaut, exposed to the actual radiation of deep space for 10 days. The chip carrying Victor Glover's cells is the first sample of a black astronaut's deep space-exposed tissue ever analyzed at the molecular level. That is not a ceremonial distinction. It is a scientific one. The human genome contains real variation in how individual bodies respond to ionizing radiation. The long-term health data used to model radiation risk for astronauts The foundational studies that produced the career limits NASA applies today was built almost entirely on white male subjects. Fighter pilots, test pilots, the people who flew in the early decades of the space program. The data was not designed to exclude anyone, but it was generated by a specific population and has been applied universally ever since, to everyone. For the first time, the avatar chips will begin to answer what individual biological variation actually looks like at the molecular level, chip by chip, astronaut by astronaut. Glover's chip is one of those data points. Koch's is another. Hansen's is a third. Wiseman's is a fourth. And whatever differences emerge between them will begin to tell us something that 5 decades of population average models never could. Whether the person standing in front of you, specifically, can absorb what deep space will do to their body for 860 days, or whether it will cost them something they cannot get back. Lisa Carnell's stated ambition: to eventually send organ chips of any astronaut selected for deep space ahead of time, test their tissue against the expected radiation environment in a laboratory, and use what comes back to personalize the medical kit they carry with them. Not a generic kit. A kit calibrated to what that specific person's cells reveal about their specific vulnerability. That future starts with these 4 chips. The Apollo astronauts could not tell us what deep space did to their cells because the tools to ask that question did not yet exist. What we have instead is a data set assembled decades after the fact, from death certificates and medical records, cross-referenced against flight manifests, and it produced a finding the field has never fully resolved. In 2016, Michael Delp and colleagues at Florida State University published a study in Scientific Reports comparing causes of death among 3 groups: Apollo astronauts who traveled to the moon, NASA astronauts who flew only in low Earth orbit, and NASA astronauts who never flew at all. The deep space group showed cardiovascular disease mortality of 43%. The low Earth orbit group, 11%. The non-flying group, 9%. 43%. Not elevated. Not a trend worth monitoring. 4 to 5 times the cardiovascular mortality of astronauts who stayed in low Earth orbit, and nearly 5 times that of those who never flew at all. The only variable separating those 3 groups was whether they had left Earth's magnetic field, 4 to 5 times higher. Kuchinada published a methodological critique noting legitimate weaknesses. The sample was small, confounding variables were real, but here is what the debate cannot settle. Those astronauts had no avatar chips. Their dosimetry was primitive by current standards. The biological changes in their hearts and immune systems during those missions were never measured at the molecular level. The Apollo cardiovascular study remains contested precisely because the question was never asked properly at the time it could have been answered. The Artemis II chips are the first time the question gets asked properly. Whatever answer comes back from those labs, it will be the first answer built on direct molecular evidence from human cells that actually went to deep space. Then consider what the NASA twin study already established. When Scott Kelly spent 340 days aboard the International Space Station, while his identical twin Mark remained on Earth as a control, researchers found that approximately 91% of the gene expression changes observed in Scott returned to pre-flight levels after he came home. That sounds like a recovery. It reads like a headline about resilience. It was not a full recovery. Not even close. The remaining 7% of Scott Kelly's genes continued expressing at altered levels 6 months after he returned to Earth. Genes associated with immune regulation, DNA repair, bone formation, and cellular response to oxygen deprivation. Chromosomal inversions consistent with ionizing radiation damage persisted in his blood cells 6 months after landing. Telomere shortening, associated with accelerated cellular aging, showed a net loss that researcher Susan Bailey linked to cardiovascular disease, dementia, and some cancers. All of this occurred in low Earth orbit, inside the magnetosphere, at radiation doses roughly half of what the Artemis II crew experienced per unit of time in deep space. 10 days is not 340, but the mechanism is established. Gene expression changes, chromosomes restructure, some alterations persist. The Avatar chips will provide the first direct molecular comparison at the deep space level, not a simulation, not an ISS extrapolation, but 10 actual days outside the magnetosphere, in the radiation environment that a Mars mission of 860 to 960 days would require continuously. A complete Mars mission delivers approximately 1 sievert, a number that exceeds every agency's career limit simultaneously. What the Avatar data will establish is whether the damage from the first 10 days projects linearly onto the damage from 860, or whether there are thresholds and individual tolerances that make some people viable candidates for that mission and others not. The answer to that question determines whether human beings can go to Mars at all under any ethical framework that currently exists. The record that will outlast every photograph from this mission is not the distance. It is this. For the first time in history, medical scientists are analyzing living human tissue, matched tissue, grown from the actual cells of the same individuals who flew, that spent 10 days in the radiation environment of deep space. Every previous window into what that environment does to a human being was filtered through retrospective analysis, indirect biomarkers, or animal models. Avatar eliminates that filter. The crew walked off the ship. They looked fine. The chips will tell us what fine actually means when you read it in the language of genes, telomeres, immune markers, and chromosomal damage compared against the same biology that stayed safely on the ground. The Apollo astronauts proved we could go. The twin study showed us what low Earth orbit costs over a year. The Artemis II crew has now given us the first direct molecular data from deep space in more than 50 years, collected with instruments that would have been indistinguishable from science fiction to the engineers who built those earlier capsules. The Apollo astronauts could not tell us what deep space cost them at the cellular level. The tools to ask that question did not exist. The Artemis II crew just sent us the invoice. Thanks for watching, and I'll see you in the next one.